Okay, so thank you again, everyone, for joining. I want to introduce Tim Chambers with the U.S. Army Natural Resource Program on Oahu. And he works a lot with Loulu and Loulu conservation. And so I'm going to pass it off to him. Tim, if you want to go ahead and share your screen, and then
Sure.
we can go ahead and get started. And then
Sounds
for
good.
the audience, you guys can go ahead and put your questions either in the Q&A or in the chat box, and we'll leave some time at the end for answering any questions.
Arissa, does that look good in terms of?
If you can do change your display setting one more
Yep,
time.
I will do that one more time. Does that look better?
Perfect.
Okay. So should we just roll with this, Arissa? We're good to go?
Yes,
Okay,
yes, good to go.
cool. Thank you for the introduction. Anyway, just repeat it. My name is Tim Chambers. I do work for the U.S. Army Natural Resource Program here on Oahu, and we operate through the Office of the Vice President for Research and Innovation from the University of Hawaii. So, in fact, Hawaii and the U.S. Army have a cooperative agreement in which they both benefit from this relationship, and that the U.S. Army Natural Resource Program on Oahu through the University of Hawaii brings capacities to the Army to manage rare resources and mitigate any impacts their training may have on those resources. And in return, the university obviously gets support in terms of funding and so forth. So anyway, I do manage the Army's, or I'm the coordinator of the Army's rare plant program. And we are not just managing Loulu, but we are directly managing a minimum of 39 taxa across Oahu. And we do that so that we are mitigating impacts the Army may have to those plant species, and in return, Army can continue to train on the landscape. And with that, I'd just like to jump into Loulu conservation in Hawaii. And this next slide is really just a collage, but Hawaii is a very unique place in terms of flora and many other things for that matter. But our flora is highly endemic and very unique in that case. And as a result, is also a little bit sensitive to human impacts and other impacts. And we also have been considered maybe the endangered species capital of the United States or the world, which is maybe not a great designation to have, but it reflects the reality and where in Hawaii, where a good chunk of our flora is at risk of extinction. And to just give you some background on our flora, we have roughly about 1,400 native plant species. Of that, about 89% of the angiosperms or flowering plants are endemic, which is a huge figure, and 74% of our ferns and fern allies are also endemic. So we have this huge endemicism in the state. But our flora is also highly threatened. And so we already have estimated 110 extinct species in our flora. And depending on how you count threatened and endangered plants, we have roughly 453 threatened and endangered plant taxa within our flora. When we look at plant species with less than 50 wild plants on the landscape, there are 204 taxa that fit this category. And when we think about plant species have that less than 10 wild plants in the landscape, we have about 103 taxa. And so, in this regard, and, I guess it leads to our reputation as the extinction capital of the United States is because 41% of all endangered species, so those are listed through the Endangered Species Act in the United States, are Hawaiian taxa. And our recent rate of extinction in Hawaii is roughly one extinction every year. And so we do have a highly unique but a highly threatened flora, and Loulu or Pritchardia species are definitely a component of those threatened species. So just jumping into Pritchardia, the genus of palms, and it's an endemic genus to the Pacific. It was first named in 1862, and there are 24 Hawaiian endemic species, and there are something like 3 or more species in the South Pacific within this genus, and potentially more. And, I'm not an expert in this realm, but, Loulu or the Hawaiian Pritchardia have been used culturally by indigenous peoples. They've been used for food, fiber, spears, weaving, lei making, shelter, like in the terms of thatching or producing, structures to protect from the elements, medicine, and they've also played a role in landscaping, and they play a role even today in the landscape industry in Hawaii. So this just gives you an idea of the Pritchardia on Oahu, and I'm focusing in on Oahu today because that is where most of our work is done, is on the island of Oahu. And when we look at Pritchardia, there are 5 species of Pritchardia across Oahu. Pritchardia kaale, Pritchardia kahukuensis in the Kolaus, Pritchardia martii, which is by far the most common species of Pritchardia on Oahu, Pritchardia bakeri, and this is last species is Pritchardia laureana. And so this kind of gives you a rough idea of the distribution of those species across the landscape of Oahu. And so just to start off, just kind of want to introduce you to each one of these Pritchardia species. And the first, I'm just going to start with the most common, is Pritchardia martii. And it exists in kolau wet forests. It has a very large fruit, and its inflorescence, or the flowering stem, are all the same length as the leaves, in a shaggy, non-spherical crown. So basically, the inflorescence is either the same length of all the fronds, and that's an important identifier for the species. The leaves are distinct in being glabrous above, but silvery gray below. And populations estimate for the species on Oahu range between 5,000 and 8,000 trees. And so, Pritchardia martii would be considered a more common taxa. It is not listed on the Endangered Species Act, but is by far the most common Pritchardia on the island of Oahu, and has its own conservation concerns. And, with the presence of coconut and rhinoceros beetle, that is one of the major threats at the moment to this taxa. So here just gives an image of the Pritchardia martii across the landscape. And then next, let's look at Pritchardia bakeri. It's considered threatened and endangered. However, it is not listed on the Endangered Species Act. It is a kolau endemic. It is a shorter tree. Its inflorescence peduncles extend beyond the crown, meaning the inflorescence itself and its stem extend beyond the length of their fronds. The fruits are rather large, and they're about 1 to 2 inches, and populations estimate split between northern and southern kolaus is about 100 plants total on the landscape. So this species is definitely of concern given its threat, but also the low number of individuals or wild individuals on the landscape. And this just gives you an idea of its habit and habitat in the Kolau Mountains. And then we look at Pritchardia kahukuensis. This is another kolau endemic, so it only grows in the Kolau Mountains on the leeward side, central to northern kolaus. It's a very tall tree, up to 7 meters, has a very spherical crown, and its inflorescences do not extend beyond the leaves. The fruits are rather small, and there's about 300 to 400 plants on the landscape currently, wild plants, that is. And this is just an example of kahukuensis, where you can look at this really spherical crown and these really tall, narrow trees. So that brings us to Pritchardia laureana, and Pritchardia laureana is endemic to both Oahu and Molokai. And on Oahu, there's about only 10 individuals, so it's very uncommon on this island. The leaf blades are glabrous both above and below. They have a large fruit, and their inflorescences do not extend beyond the leaves or the fronds of the crown of this tree. And then finally, this brings us to Pritchardia kaale. This is the only federally listed critically endangered taxa on Oahu. And it has a small fruit. Its inflorescences extend way beyond the crown. And you can kind of see here in this upper corner, let's see here. Excuse me for a moment. I'll show you in a minute, those fronds. And the population estimates there are about 1,300 wild plants growing in the Waianae Mountains. And again, this is a Waianae endemic taxa. And we have outplanted or reintroduced plants. There's roughly 683 plants on the landscape. And I just want to point out this is the one species of Pritchardia that the U.S. Army Natural Resource Program manages. The other taxa are managed through DOFA and the Plant Extinction Program on Oʻahu. And so this plant is, like I mentioned, federally listed, but it also has received maybe conservation actions beyond the other taxa, in that we have begun to outplant or reintroduce this plant on the landscape. Where those other taxa, there are no reintroductions representing those species as of yet on Oʻahu. And so this plant has quite a few wild individuals, but those outplanted individuals are growing the population on Oʻahu through management efforts. And so just this example of Pritchardia kaale's habit and habitat. And here you can see the fruits dangling below the crown, which is just an example of those inflorescence greatly exceeding the length of the fronds of the plant. And that's an important identifying characteristic for this species. So Loulu has many threats, and I just want to point those out. So obviously invasive species are problematic for this taxa. Ungulates, both goats and pigs. Goats and pigs are damaging, in some cases feeding on the plants, but in other cases damaging the habitat these plants need or rely on to survive. Rodents, rats largely, however, mice probably have some impacts as well, do damage to the plants directly, but they're also predating on the fruits and eating the seeds. And so they can really reduce recruitment or future generations by consuming many of the fruits that would otherwise maybe land in the landscape and produce new plants in the wild. And then, of course, invasive species in terms of weeds or plants also are competitive against a Loulu in their landscape and require removal to produce a better habitat, so survivorship is higher for these, the plants in the landscape. And then, of course, these broader threats such as wildfires, landslides, or the ominous climate change, are also directly impact this species in the wild, and not always necessarily easy to mitigate for, but those are direct threats to this species as well. And so that brings us to the new threat, the Coconut Rhinoceros Beetle. I don't want to belabor the history of this invasive species on the island. However, it was first detected on Oʻahu in 2013 as native to Southeastern Asia. It's been introduced to roughly 30 islands throughout the Pacific. And its host plants right now, to our knowledge, are largely palms. That includes Loulu, and then maybe secondary host plants such as banana, hala, kalo, and sugarcane. And so just to give you an idea what the CRB damage looks like to these plants, we can see here some examples. Over here, you see these V-shaped cuts caused by CRB. This is a coconut palm, not a fan palm. And so you can see these V-shaped cuts. You can also see the presence of boreholes where those beetles have entered the plant. And we see the V cuts, and then, or another sign would be these scalloped edges. And really in fan palms like Loulu's or Pritchardia, they are all fan palms, so largely the damage on the fronds are expressed in these scalloped edges that are exemplified here. And then if we just move on, here are some other examples from the field where we've identified CRB damage on Pritchardia kaale. So here is borehole through the spear, which is ultimately new leaves tied up, ready to expand, where the beetle had entered and fed through. And the second image here is actually the stem of a Loulu that was killed by CRB, and we split it open to investigate the internal damage. And we see that, basically the Loulu entered through the spear, consumed the meristematic tissue, so those growth points of the plant, and have, further eaten through the Pritchardia stem to the end. And so this is another example of a borehole in this image. And then on this side, we can actually see a plant that is going to succumb because of CRB feeding, where the meristematic tissue was fed on and consumed by the CRB beetle, and the only thing remaining alive are kind of the lateral leaves. And this plant has nowhere to grow into the future, and so will eventually succumb to the damage inflicted on it by the CRB beetle. So now that I've kind of introduced, the threats, the species that we have on Oahu of Pritchardia or Loulu, I'd just like to talk about some of our conservation efforts to manage these species in the landscape and try to stabilize populations so they are not quite at risk for extinction. And there are two kind of general approaches to conservation that we employ for this tax. So the first would be ex situ conservation. This really means off-site conservation. So it's conservation that's happening outside of the wild context. And as you can imagine, that includes field collections of Loulu to be brought in so that we can preserve them off-site. And we can do that in multiple ways. We can do that through seed banking. We can do that through tissue culture and micropropagation, and we can do that through maintaining living collections. And that could be in containerized living collections in a plant nursery or living collections at a botanic garden. And so that is basically safeguarding those genetic resources, safeguarding some of those wild founders off-site in case something in the wild were to happen to further threaten those remaining individuals. And so we generally referred to that as capturing genetic storage. And so that when we need propagules to go reintroduce a population or put plants back on the landscape, we have a source of those plants to do it. But it's also hedging our bets and kind of a insurance policy for keeping these things away from extinction. Because if we have seeds in a seed bank or plants living in tissue culture, or in a living collection, we can always propagate those things to establish new populations into the landscape. And so here's just kind of the flow chart of ex situ conservation from field collections to processing that material, putting it in your storage, either a seed bank, tissue culture, or living collection. And then when reestablishing populations, we want to pull that material out, propagate it, and return it to the field through outplantings or reintroductions. The second approach would be in situ conservation, and that's conservation of these resources in their wild context. And in doing that, we want to, in situ conservation is really important that we secure those wild populations and begin to control those threats that threaten them in the landscape. And we do that in multiple ways. So controlling threats in terms of habitat, we are building big fence enclosures, which many of you might be aware of, in which we can then remove the ungulates, set up rat grids to trap the rats that may be impacting those plants, and then securing those populations through enclosures, and then the establishment of threat control, either through invasive species control that may use some chemical or physical removal, rodent or rat trap systems that are going to control those impacts on those plant species. And other methods of conservation, of course, monitoring those populations so we know where they are and what their status is, surveying new areas, so finding new occurrences or new populations of these plants, threat control that I mentioned before, and then, of course, outplanting or reintroducing populations to help stabilize these plant species on the landscape. So with that, I just want to kind of jump into, a case study of conservation. That would be Pritchardia kale, because that's most of the experience I have managing these species. And through the Army, we have converted our biological opinions into implementation plans. And those implementation plans outline how we should approach or provide a framework for how we should approach rare plant management for the taxa that we're managing, or stability of those populations. And so in doing that, we have some basic requirements that we have to meet for our management. One is we need to maintain 3 populations, which we call managed for stability populations. And that is, we're managing 3 populations where we're controlling all the threats, and then we're looking to create genetic storage or make collections of those wild plants that we can represent off-site, either in a seed bank, tissue culture, or living collection. We want to manage populations of those 3 populations that have at least 25 reproducing individuals in each, and this is due to its long-lived nature. And we want to create a situation where we have stable population structure. So if we outplant 50 individuals at that population or that reintroduction, we want to manage things so we start to see the building of new generations. So stable population structure will include having not only mature plants, but immature plants and seedlings on site so that we see multi-generational populations start to develop where we're trying to recreate populations. And then, of course, that final requirement for stability is complete genetic representation of all population units we manage in some sort of storage. Again, that could be seed bank, tissue culture, cryopreservation, or living collection. And complete genetic representation generally would be securing 50 individuals from each population in storage, or all individuals if less than 50 in a population. And the many challenges we face are really just controlling those threats, obtaining access to these plants. These plants are definitely located not in the easiest places to get to, so we need to be able to use ropes and rappelling and tree climbing along with helicopters to access these populations. And the seeds don't store well, so they don't store in conventional seed banking conditions, which is roughly -20 degrees centigrade, and seeds dried. Well, the seeds of Pritchardia would be defined as exceptional, quote unquote, and exceptional species are those species that ultimately do not store well in conventional seed banking conditions or have other issues. They don't produce seeds or things like that. And in the case of Pritchardia, we are working right now with Lion Arboretum to build protocols to understand how to secure seeds or the embryos of those seeds in cryopreservation, so at a -194 degrees centigrade, to serve as a long-term storage option for this taxa, because as of now, we can't store it in a conventional seed bank. We can store them as living collections, but that becomes more dangerous since CRB is present on the island and they are a threat from that invasive species. So we are currently working on protocols to secure these things in laboratories versus in living collections so that we can preserve these species that are highly threatened by Coconut Rhinoceros Beetle. So right here is the current distribution and historic locations of Pritchardia ka'alai. I just wanted to point out that we have a mixture of wild populations and reintroductions throughout our management systems, and its range is pretty small, as we saw before in this small portion of the Waianae Mountains. So just to give you background on the current status of Pritchardia ka'alai, it has been before the onset of Coconut Rhinoceros Beetle and knowing all the potential threats and watching the potential threats of this beetle on resources like Hawaiian Pritchardia, this species was really serving as a poster child of stabilization for Hawaii, because what happened is once we remove those threats to the plants, like fencing populations and removing the ungulates so we can maintain good habitat, controlling rat populations so seeds were actually finding their way into the forest floor and could germinate into seedlings. We conducted all that control and we saw the impacts of that control, which was population building. We started to see seedlings develop, and those seedlings matured into immature plants and then eventually mature plants. So through those control efforts, we were really able to start to see the positive impacts of stabilization on this population. But I think CRB has thrown that a little bit into chaos, given as it will have huge impacts on our Pritchardia resources. So right now, our program has established 5 reintroductions with all threats controlled except CRB, but I'll get that into a little bit later. And currently those reintroductions maintain 148 mature plants, 529 immature plants, and 6 seedlings. And when we look at the wild populations, those numbers are also looking really good. And those populations in part are protected by enclosures. And so currently we see 205 mature wild plants on the landscape, 634 immature plants, and 477 seedlings. And just to give you an idea how maybe CRB is impacting both these reintroductions and wild reintroductions, since we've seen the CRB beetle kind of move into the montane landscape and move into higher elevations. In wild populations, we've observed 11 plants damaged and 1 dead plant. And at reintroductions, we've observed 27 damaged plants and 2 dead plants. Now, I will throw in one caveat for wild populations. Some of those occurrences are really hard to access, so we're not monitoring all that frequently. And in some cases, we survey them by helicopter. And so we do have a little bit more survey work to do to kind of understand where some of these wild plants that are harder to access, how they're being impacted by CRB. So what do we do today in the face of CRB? So how do we protect Loulu in the face of Coconut Rhinoceros Beetle? So as you know, we are in early stages of trying to understand how to control this beetle, especially in kind of the forested system and not the urban context of Oʻahu. We are limited with the tools to control the beetle and in Loulu's wild habitat. And part of that is because, you know, some of these controls are chemical and those chemicals are generalists, and we really have to be careful on how we treat Loulus and what those non-target impacts would be to other arthropods or native insects that are utilizing or interacting with Loulu species. And so we have to be very careful about implementing, and in many cases, those chemicals aren't labeled for use in a forestry system. So as new tools of control are developing, we have to find ways to safeguard these in the interim. And the way we've selected to do that on Oʻahu and that's gaining traction across the other islands is securing living collections. So this genetic storage, this idea of genetic resources being maintained off-site, but in this case, outside of Hawaii, where there is no presence of CRB. And that really has a lot to do. That decision has been made because we are really limited in our control mechanisms at the moment. And so we are targeting 50 to 75 individuals of each species and 3 to 5 individuals per wild plant, except for Pritchardia lauriana, and that's because Pritchardia lauriana has just so few individuals on the island. So the idea is we want to go collect a representative sample of seeds from across these species populations that can be then established as living collections in partner institutions. And right now we have 2 partners working with us on this, the San Diego Zoo and the Montgomery Botanic Center. And we, of course, are looking for new partners. So basically, we are sending them live palms to be established in subtropical areas throughout the continent at the moment to safeguard these species. So in the case that CRB were to extremely damage the populations and plants on Hawaii on Oʻahu through time, that we can return to those living collections for propagules or fruits that we can propagate and reestablish populations on Oʻahu when more control mechanisms are available. So, but that is no small task. As I mentioned, access is a big challenge. And so in order to you be able to collect from these trees, especially for the 4 taxa not managed by the Army has a bit of an easier task because we've established representation of these wild plants in many places. So we have more locations to go sample seed from. But DOFA-PEP has a great challenge. They have to go find these remote resources, a lot of times through helicopter work. There's got to be a lot of gear and training, so people have to be trained in rope access, whether that's climbing trees or rappelling off of cliffs. And here are some examples of collection efforts through DOFA-PEP, using either having direct access to fruits on the ground, using extendable pole pruners to pull fruits out of the canopy, or of course start to use our trainings in tree climbing and rope access to access those fruits and trees in the landscape. And so to close off my presentation, I just want to kind of throw up some of the fruits from a handful of the Pritchardia that we have on Oʻahu. And with that, I wanted to give a shout out and acknowledge both Josh Serrano and Susan Ching for the support in developing this presentation, especially through PowerPoint slides, images, and background information on those Loulu species that the Army has little experience managing. So thank you very much.
That was wonderful. Thank you. For everyone that's joining us today, please feel free to share any questions in the chat or the Q&A. Looks like we have some coming in. We have a question from Jonathan. What are the natural pollinators and mechanisms of seed dispersal?
Well, I guess what are the natural pollinators? So natural pollinators, I'm not exactly sure. I mean, they're insect pollinated. They're small flowers, a huge amount of flowers in an inflorescence. They produce a lot of pollen. So do we know the exact specific pollinators? No, but I think in general that's insect pollinated. And we're trying to understand, especially in the light of CRB and, really relying on chemical defenses to manage CRB, we have to be concerned about the native or endemic arthropods or insects that are using Pritchardia. And so there are some investigations underway through the Army with some of the researchers that collaborate with us to look at eDNA on Pritchardia and to kind of understand what kind of arthropod species are utilizing, get a better understanding of what species might potentially be pollinating Loulu. And then mechanisms for seed dispersal is a hard one. I think right now the biggest disperser for Pritchardias is gravity. And there may have been some avian species that were in the past that existed on Oʻahu that may have been distributing that seed. This is the background that I can't be totally confident on, but it's possible that some of these rats that are eating the fruit are moving these fruits around or dispersing the fruits, but I think in general, research would suggest that's probably very limited because most of the rats' interaction with those fruits are very destructive. So my guess right now would be dispersals are really reliant on gravity in the landscape.
We have another question here. What are some of the challenges to Loulu propagation you've encountered? Then we have some comments as well.
Okay, interesting. So propagation, so challenges in propagation. Well, one, I think Pritchardia is challenging in its seed storage behavior, right? It's we kind of have orthodox seed on one end of a continuum here, and those orthodox seeds tolerate desiccation. They survive well at temperatures at around, once desiccated, then maintain viability when stored at temperatures around -20 degrees centigrade. That's orthodox seed, so those are like prime candidates for seed banking. On the other end of that spectrum, you kind of have recalcitrant seed, and those seeds, recalcitrants meaning difficult, generally need to maintain a high moisture content to survive, and therefore we can't desiccate them without killing them to the point where we need to remove free water within those seeds so that we can freeze them so that those cells aren't damaged through the freezing process. And because we're unable to desiccate them, they will not store at the -20 degrees centigrade. However, as I mentioned before, we are looking at research to preserve at least embryos in cryopreservation or at liquid nitrogen temperatures in -194, 96 degrees centigrade. And Lion Arboretum has made some good progress with those protocol development. We're just not right where we are. We're not right we don't know as much as we need to know quite yet to actually rely on that technique for storage. So what you will see for Pritchardia is we don't have a lot of genetic storage, right? We rely on those wild plants. And so propagation, as long as you're working with fresh seed, seems to be pretty straightforward. You know, generally we propagate these in a moist media in Ziploc bags, and we'll incubate them at like 25 to 30 degrees centigrade. And germination is pretty reliable. Now, germination gets less reliable the longer these things are stored or sit around. Um, And also from propagation, you know, looking at tissue culture and micropropagation, we've also used micropropagation as a technique to propagate these species, especially when we go into access plants and all that's available is immature seeds. And so we can use techniques in micropropagation to germinate those immature embryos and then produce a plant. So that's actually one of our strategies when making collections so that we can move living collections outside of the state to safeguard them in the absence of CRB is utilizing micropropagation to do that. To just get back to the question of soil pH, I think soil pH has a huge role to play in, you know, in rare plant management, kind of understanding the conditions under which we propagate these things successfully, but also where we return to them to the landscape to be successful. And, you know, in our program, in generally, I think we lack a lot of that soils data. So it's really interesting to see the differences in pH and then maybe plant performance and outplanting based on a pH gradient. And so I think that's an area that really needs more investigation as we are looking at the limitations of our rare plant species, and Pritchardias or Loulus included in that, you know, so that we can identify appropriate areas for reintroduction or, you know, for successful reintroduction. So.
We have another question here. Any evidence of hybridization among the different Pritchardia species?
I'm definitely not an expert in this realm, and but I think it's safe to say that these taxa are highly likely to hybridize with each other when brought into close proximity. So if you're maintaining living collections somewhere with multiple Pritchardia taxa, and we want to minimize, you know, when going back to those living collections to get seeds for reintroduction purposes, we definitely want to minimize that hybridization. And so it's been, I think, pretty clear that if we're managing these things in living collections, then we have to use techniques that isolate them other ways so that we're not getting hybrid seeds when looking to restore these populations. So I think there's a lot more information to learn there. There's been a lot of observations of potential hybridization. And I do know at the University of Hawaii, working with Lion Arboretum, they do have a master's student that's kind of looking into that question of hybridization, but then also how do you isolate these things in conservation collections as to not bring in hybrid materials into reintroduction. So I think there's ongoing work, and as we rely more on living collections as our ex situ conservation approach, then we really have to learn more about how we utilize those resources effectively, minimize hybridization, all that kind of stuff. So these, I think, will be ongoing areas of investigation into the future to kind of understand that better.
Great, thank you. I think I'm not seeing any more questions, so I think we can go ahead and end this. I just wanted to say thank you again for sharing all that information with us. It was wonderful and really fascinating to learn about. Learned so much today, so thank you again, and thank you everyone for coming. Again, this recording will be up on the YouTube channel. I shared the link in the bio or in the chat box, but yeah, thank you.
Yeah, thanks all for having me.